Yay, starting. Cool. Welcome. We are getting close to the end of metabolic diversity unit, which means we will soon start doing reviews. Did somebody, I believe many people already looked review slides that I posted for unit 1. So if I have time today, depending on how we feel, like if I see lost faces around, I will let you go. But we might have a time to go through a review session. Although everything is written there. So I got a few questions on what will be on quiz. So as you probably know, I cannot really tell you what will be on quiz in terms of questions. That would be really stupid, right? Yeah, I mean, I can lead you through that, but if I tell you what will be on quiz, what's the point? I give everyone A+ and we go for a beer. So I know that would be ideal, but then I lose a job and yeah, that can't happen. Anyway, so it won't be hard. And as last year, nothing will, I mean, I will change some questions, like I will definitely add more and reduce something, but it won't be hard. So don't panic. Like quiz will not be like my goal is not to take marks from you. My goal is for you all to get great marks. That's all. I have no intention to fail you. And I'm like, you can ask from last year, whoever took the class. I am really prepared. So let's dig in. Let's dive into the metabolic diversity. This thing doesn't love me. Yeah, okay. So we covered phototrophy, chemolithotrophy, and fermentation in previous 2 weeks. Like actually two classes, 1 week. So today we are going to talk about anaerobic respiration, and on Thursday we will end up with hydrocarbon metabolism, my favorite. So I'm thinking that maybe on hydrocarbon I will have more time to go over reviews, and then I will try to cover both sessions because that will all go on your quiz one. Which is coming up. Yay! First quiz. So when we talk about anaerobic respiration, the first thing that comes at least to my mind is this picture here, which is called Tower of Power. And again, there will be always questions, should you memorize this? Of course not. But you do need to know how to read Tower of Power. And as we are going down into the anaerobic respiration and its diversity, we are going to learn and understand all points of this. Now it looks something like many colors picture. So, anaerobic respiration, which maybe sounds weird because whenever we say respiration, everyone associates that with oxygen. So how is that anaerobic? Well, the anaerobic respiration literally means respiring something else, something other than oxygen. So something else is going to be terminal electron acceptor. And that something else is placed on this tower of power. So anaerobic respiration is usually opposite of highly efficient process of aerobic respiration, because aerobic respiration relies on oxygen, and as you can see, oxygen is very down on the tower of power, which means it has highly negative redox potential. So, oxygen actually has the more active, highly positive whatever is down on the tower of power has higher affinity to accept electrons and to be terminal electron acceptor. And the best terminal electron acceptor is oxygen. So oxygen on the bottom, whatever is on the top is not good electron acceptor, but it is good donor, which is why you see glucose on the top, which means whatever is on the top has a lot of energy, which means it's full of electrons that can be taken from the molecule. So molecular oxygen is the molecule that's changed the world, well, actually shaped the planet in the way how it exists now, and it has high affinity for electrons, which means it's the best terminal electron acceptor. That's why we say it has highly positive redox potential. However, we learned till now The first organisms that lived in this planet actually did not have available oxygen, because we know that oxygen was made by cyanobacteria after banded iron formations, after all iron was exhausted and we saw banded iron formation. But the bottom line is that first metabolism was not related to oxygen. So anaerobic respiration is much older. So anaerobic respiration is related to alternative electron acceptance, something that is less suitable to take electrons, to accept electrons, so less positive than oxygen. And we will cover a few of terminal and electron acceptors. So some of them we already mentioned when we talked about different type of organisms. So it will be a lot of overlaps. So the first one is nitrate, NO3 minus. And you will see me using NO3 minus often, simply because I am constantly messing up the pronunciation between NO3 minus and NO2 minus. They're different compounds, nitrite and nitrate. So NO2 minus and NO3 minus. And they're different molecules and different terminal electron acceptors. So that's why I tend to use chemical formulas, because that's more precise. Each organism has to have energy source. So each microorganism has to have something that will be full of energy, aka electrons, and it will use that molecule that is highly reduced, full of electrons, to extract those electrons, which are energy. Some of the first molecules that we covered on early Earth are hydrogen sulfate, hydrogen, iron 2 plus, and it's important to say it's iron 2 plus, which is a ferrous iron, because that iron is less positive than 3 plus. And why it's important? Because one that is 2 plus can release electrons, and you will see that in a few minutes. Microorganisms simply evolved, or to say in a lay language, learn how to extract electrons from anything that was available on early Earth. And iron was available. Hydrogen they were extracted. They used it to extract electrons. I'm apologizing. I have to restart my recording because my Zoom, for some reason, just... Okay, it's recording still. Anyway, if it's not good, I will pause from last year. So extracted electrons are then delivered to alternative electron acceptors. So alternative electron acceptors are nitrite, nitrate, sulfate, or carbon dioxide. And we know who is fixing carbon dioxide. So carbon dioxide is the electron acceptor. And surprisingly, proton, 8 plus, can also be used as a terminal electron acceptor. And there is one very specific organism that we will mention at the end of class. It has a very interesting name. It's called Furiosus, but you will see it later. It's very interesting, and I'm thinking it's pretty unique. But let's start with nitrate, which is nitrate distillation. So if you look at the tower of power, oxygen is far bottom. NO3 minus is slightly above, which means that if you have to choose of all available electron acceptors in this world, the best one would be NO3 minus if you don't have oxygen, because it's so far down on tower of power. So it's decently good. So you can decently good, it means you can generate a lot of energy from respiring NO3 minus. So in order to respire NO3 minus, that means NO3 minus will be reduced, something has to be oxidized, which means something has to be used to extract electrons or energy from it. So there are two modes of nitrite reduction, assimilative and dissimilative. And they're different, they're used for different purposes in cell. So if reduction of NO3 minus is dissimilative, that means that the NO3 minus is required to create energy, to create proton motive force. On the other hand, if reduction of NO3 minus is happening for assimilative purposes, that means that reduction of this compound is generating certain organic molecules that are assimilated into the organism and used for biosynthetic purposes. And this nitrate respiration, NO3 minus respiration, is going in the direction that is presented here. And purposefully, I'm putting brand names and chemical formulas. I prefer chemical formulas, but you are very welcome to memorize only names. So nitrate respiration goes from nitrate to nitrite to nitric oxide, nitrous oxide, and denitrogen. So you can literally see how that respiration goes from like losing one oxygen, and very often this respiration ends up with nitrous oxide, which is N2O. There are certain microorganisms that form nitrous oxide reductase, and that means the enzyme that, so each of these conversions from NO3 minus to 2 minus to NO to N2O and N2, each of these steps is catalyzed by specific reductase. So it's nitrate reductase, nitrite reductase, nitric oxide reductase, and the enzyme nitrous oxide reductase is awesome, and especially because it can convert N2O to N2, the nitrogen, and for those who don't know, N2O is a very highly potent greenhouse gas. It has 200 times more potent than carbon dioxide. So if you have an enzyme that can reduce the amount of N2O, you are blessed. So, nitrate reduction is simply called denitrification, which means getting rid of NO3 minus. So the process of denitrification is very significant for agriculture, and for agriculture, it's actually not good. It's detrimental. Why it's detrimental? Because denitrification means getting rid of nitrate, and nitrate is a food for plants. So if you have denitrification organisms, you are damaging crops. But denitrification enzymes are beneficial for wastewater or sewage treatment, because in that wastewater you have a lot of nitrogen compounds that are waste. Many of them are NO3 minus, NO2 minus, not so much gases. Well, there are, but mostly those that are soluble. And denitrification enzymes can help in detoxifying wastewater. So it has good and bad sides, as everything in life. So microorganisms that do denitrification from phylogenetic bacteria. And later, so when we talk about different different levels of diversity, we will talk about metabolic and functional diversity. But later on, we will move to the diversity of bacterial phyla and archaeal phyla. And you will see that among bacteria, the most abundant and the largest phyla is proteobacteria. And denitrification organisms are part of that group. Physiologically, they have facultative virus. That means that even though they prefer anaerobic respiration and they love to respire NO3 minus, if you expose them to oxygen, they have ability to adapt and respire oxygen too. This is opposite from some other types of, we call them obligate anaerobes. Obligate anaerobes are organisms that would die if you expose them to oxygen. These facultative aerobes will be fine. We will talk about, there are also facultative anaerobes, which E. coli is. We will talk about that later. So denitrifying bacteria are metabolically very diverse. What that means? That means that even though they respire NO3 minus, they can extract energy, those electrons that I mentioned, from variety of organic or inorganic compounds. So metabolically, they're very diverse. So they are anaerobes. They do anaerobic respiration, but in terms of energy, they will take electrons from many, many stuff that you give them. And that's why we say they're metabolically very diverse. Archaea can also use nitrite. One of the interesting features, and I will mention that in next few slides, is that even eukaryotes are known by the ability to respire NO3 minus, And I said that, I know I said that I'm not going to cover eukaryotes here, except those that are microbes, but these things sounded very interesting to me, which is why I'm going to mention this bending foraminifer. God help me, what is that? Some sort of animal. I never saw it, but physiologically it's really interesting. Imagine this, even though it's an animal, it's an eukaryote, it has a lot of nitrate in cell. So it was found that these, whatever they are, animals, I mean, they are eukaryotes, so they're not bacteria, they produce dinitrogen. And dinitrogen production used to be thought that it's typical prokaryotic feature, but apparently it's not. So in this organism, they're like, okay, there must be some symbiosis with identifying microorganisms, but they didn't find anything interesting. So imagine where denitrification is happening, even if it's not written on slide, it happens in mitochondria. So even though there are no known denitrifying organisms living there, basically mitochondria are bacteria. So, I mean, they were ingested long time ago with endosymbiosis. But that's where denitrification is happening in this benthic foraminifera. So they don't have oxygen, but they still live. So the reason why I'm mentioning this is because even though it's not, like there are no isolated denitrifiers from this organism, the whole process of denitrification is still happening in mitochondria, and we all know that mitochondria are bacteria. So definitely there are prokaryotes associated with this organism. That's the only reason why I'm mentioning. The other example of eukaryotic denitrification is Fusarium, which is, of course, fungi. So even though it's not really organism, it's not microorganism, it's not micro, you can see kind of little hairy roots, we still think they are micro-sized. Again, mitochondria, so bacteria associated with this organism are converting NO3 minus to all other nitrogen oxides, but they can still use oxygen. So that makes them, so this is called hybrid respiration, but and both processes are occurring in mitochondria. And now this makes me wonder, are they facultative aerobes or anaerobes? Which one would they prefer? What do you think? Yes, why? Perfect. Yes, excellent. Because the oxygen is more efficient. It's far down on tower of power. So that's like a glucose lactose in lacopero. So the one that will give you more energy, you'll take that one if both are available. But once when you don't have that good one available, I mean, what the heck? You can take whatever, you will take whatever is there. Literally. And that's why they're so cool. They can do whatever. The other alternative electron acceptor. So we are not anymore at nitrate, we are here in nitrite, NO2 minus, which is a second step after NO. So when NO3 minus is reduced, it loses one oxygen and you get NO2 minus. Now, who is doing it? Anamox and this beautiful organism that is called, I call it Oxifera because I cannot pronounce Methylo mirabilis. That it's kind of, I have a hard time pronouncing Latin names. I memorize them, but I can't say them. But physiologically, Brittany will probably tell you that the first one is more interesting physiologically. I'm personally a big fan of Oxifera, and there is a reason why, because that was the way how when I did my first response to reviewers, I actually used my knowledge of this organism to sound very smart. So that's why I love it so much. I still feel smart when I remember that. I mean, there is a full story about it, but I will tell you when we have a coffee in front of the remedy, when we look at your quizzes. So this Anamox, I mean, we mentioned it. You remember that we talked about Anamox, right? They are very famous anamoxosomes, and we will go through them again. That's that repetition that I was telling about, and you can already conclude what am I going to ask you in quizzes. So the energy source is what? Ammonium. What is carbon source? carbon dioxide. There is no carbon involved in oxidation here. So terminal electron acceptor here is actually nitrite, NO2 minus. And these guys actually developed something called anamoxosomes, which we call pseudo organelles. They're not real organisms. I don't like saying that prokaryotes have them, but they are surrounded by ladder and lipids. And these lipids are quite significant simply because they prevent toxic metabolites to be released into the cell, because when cell is respiring NO2 minus and oxidizing ammonium, it has intermediate products like dinitrogen, which is not toxic, but this hydrazine is extremely toxic. So if they don't have those ladder and lipids that surrounded all metabolic pathway, they would die. And finally, Candidatus mirabilis toxifera, or how I call it toxifera. So beautiful methanotroph. A very beautiful methanotroph, which means it is oxidizing methane. And don't get confused with methanotroph versus methanogen. Methanogen is the organism that produces methane as a part of metabolism. And we talk about methanogenesis as one of the primary metabolism on Earth, and they have that very interesting carbon dioxide fixation or acetylcholinase pathway. This is methanotroph, which means it degrades methane. So methanogen means it generates it. The troph, methanotroph, means it eats methane, so it tops it up. So methane here is carbon source. Electron accepted for oxygen is nitrate. And why is that so cool? Because it is anaerobic. And why it's anaerobic? It's anaerobic because it doesn't respire oxygen. And here is where confusion comes. This is where I sounded very smart. So what is this organism producing while oxidizing methane and respiring NO2 minus are nitrogen and oxygen. There is oxygen involved, right? But does that mean that this is aerobic? Nope. Why? Because the oxygen that they make from oxidizing methane and respiring NO2 minus, that oxygen that is built up internally is now going back and it's used to activate other methane oxidation enzymes, like just to keep it going. So even though there is oxygen involved, this is not aerobic. It does not respire methane. It does not respire nitrite. So that's where I sounded very smart, because one of the reviewers said that I'm wrong about like saying that my methanotrophs are the only thing because they are aerobes. I was like, haha, no, you are wrong because you don't know. That's how I learned that I cannot really let my Serbian out when I am giving response to reviewers. Yes. It cannot respire it. It's literally obligate anaerobe. It makes oxygen, but the oxygen cannot be respired. It cannot use oxygen to generate proton motive force. It has to have NO2 minus. It will make oxygen to activate methane monooxygenase, but it doesn't respire. It would die in their face. I loved it. I still remember that I was young grad student when that happened, and I will never forget it. This organism belongs to NC10 phylum of bacteria, and it's the only one that I know. Third one is sulfates. And I love these organisms because when something, so if you think of tower of power, so oxygen is on the bottom, then you have, so this is where oxygen is on the bottom. Slightly above is a nitrate, then nitrite. And sulfate is really in the middle. So on the top will be glucose, right? This sulfate is somewhere in between. So SO4 2 times minus, or elemental sulfur, which is S0. So they're not the best terminal electron acceptors, but they could be used. Like if you have nothing else, of course, that's what you have to use. So and again, there is a very good reason why I love them. They're important and for biotechnology and for many other things. So this sulfate is terminal electron acceptor. You can see where it is. So it's here on the orange part here. So labeled here. So it's what we say is a mild base for energy. That means it's not really bad, but it's not the best. Like you can use it, but it's not really going to be used for it's not going to generate a lot of energy. But if you can couple sulfate respiration with something very full of electrons, something very good for energy, then you can generate a lot of protein motive force. What that means is that what can be used to extract electrons must be above. So it has to be part of, it has to be highly redox negative, because that has to be coupled with sulfate respiration. So if you think about that, if you couple glucose with oxygen, the most energy is extracted. But if you couple glucose with sulfate, it's much low, it's a very small distance. So when electron travels, it releases protons, that's creating energy. So you have very tiny distance to travel, so not a lot of energy. That's the point. So what that means is that literally bacteria organisms that are able to respire sulfate can really only use glucose or hydrogen. So again, in ancient times, glucose was not available, so it would be hydrogen. And this is something that, so this is that proton motive force that I always talk about. So when oxidizing a fuel source and reducing a sulfate, so microbes are able to create this H2S, hydrogen sulfide. And where did we see hydrogen sulfide in tests? We saw it when we talked about anoxygenic phototrophs. We talked about hydrogen sulfide that is electron donor for phototrophs. Now here you actually have completely different metabolic group where part of metabolic production is that hydrogen sulfide. So they're respiring sulfate and make sulfide. So that's why we call them very creatively sulfidogens. They make hydrogen sulfide. So they will be common in soils, in marine environments, simply because that's where all sulfates are abundant. And I really like the name of SRB, which is sulfate reducing bacteria. Simple reason why I like SRB is because that's the abbreviation of my home country, so I will never forget. That's how it says in my passport, my old passport, SRB, sulfate reducing bacteria. That doesn't mean they're from Serbia. They're just there. They don't need passport. They can go without. Anyway, so in order to have this sulfate respiration going, you have to have something highly reduced, which is a hydrogen, which is organic compound. Probably in ancient time was hydrogen. There was no organic compound. So producing bacteria that are a lot are relying on hydrogen, and they produce sulfide, and usually they are in competition with methanogens, organisms that make methane. So if you think about that later on, when we cover, actually I think it will be next week when we cover methanogenesis. So methanogens also use hydrogen as energy source. That's why they're ancient organisms, but they're competing. They use the same one, hydrogen. Of course, SRBs can use organics, methanogens can't. But in terms of terminal electron acceptor, sulfate reducing bacteria use SO4 2-. Methanogens are using carbon dioxide. So there is a question who would win. Again, it really depends on what is present over there. So if they're called sulfidogens, that means the process that they are performing when they make hydrogen sulfide is called sulfidogenesis. And usually this sulfidogenesis, you will recognize it's very stinky. It stinks like a bad eggs. that's hydrogen sulfide. And that happens in the areas where organic matter is concentrated. So why? Because those highly concentrated organic matter area, they're loaded with sulfates, like toss of trash. So that's where those sulfidogens actually outcompete methanogens. So even though that's maybe good for environment, it really stinks because there will be a lot of smell. So when you dispose sewage sludge or garbage in the ocean, that will actually increase sulfidogenesis because there are many SRBs over there. So whatever compound you toss over there is full of sulfates, and they will just respire it and make hydrogen sulfide. And oil sands are another source of sulfides. So that's where you will, if you want to do enrichment for sulfidogens, that's where you will find them. And similar like nitrate reduction, sulfate reduction can be assimilative and dissimilative. Again, if it's dissimilative, that sulfate respiration, it's done for the energy and it creates hydrogen sulfate. If it's assimilative, that is the process that is basically means making sulfur compounds that will be assimilated into the metabolism. And there are some amino acids that contain sulfur, so it's literally building a building block for the organism. So one of the examples where sulfidogenesis is highly important is on the metal surface, usually on constructions of bridges, because microbes that are located on that metal surface will consume more oxygen available. And when the oxygen becomes depleted, the environment becomes anaerobic. Now, when the environment is anaerobic on those metal constructions, that's a fantastic place for sulfate-reducing bacteria to come. And when sulfate-reducing bacteria come to those now anaerobic metal surfaces, they will respire sulfate and they will make hydrogen sulfide. Hydrogen sulfide can react with metals and usually with iron. And those so-called iron or metal sulfides will cause corrosions of bridges. So that's why metal constructions on bridges are coated to prevent biofilm formation, because that's how they prevent those corrosions. It's one interesting fun fact about sulfate-reducing bacteria. Sulfate reduction is also very interested in symbiosis. So this is why these organisms really fascinate me. Like sometimes you can't believe that something like that actually exists. And that means, so those organisms form consortium, which means they cannot live without each other. And they are simple called or anaerobic methane oxidizing archaea, or anime. If somebody is watching those cartoons with, like, I forgot the name, but my husband is watching, it's called anime. So that's how you memorize it. Like, I don't watch it, but he does. He's obsessed with that. Anime are archaea. These archaea are methane oxidizing organisms, which means they are analog to methanotrophs. They use methane. So they consume methane. But they cannot live alone. They need these sulfate-reducing bacteria. So they're connected to each other. They're literally like BFFs forever. So anime is using methane as a carbon source. That's all cool. And that's also an energy source. But they respire sulfate. On the other hand, sulfate-reducing bacteria are also respiring sulfates, but they're using organics. So they don't take carbon source from anime. So what sulfate-reducing bacteria are doing for methanotrophic archaea is creating an energy. So while they're respiring sulfate, because that's their normal procedure, they're creating this energy molecule reduction equivalent NADH. So they are making that NADH and they're giving it to the anime, so they're required for each other. And And a reaction is energetically favorable, and it's happening only in this syntropy between ANME and SRBs. So this, for me, this is very interesting, and I don't think ANME was ever isolated alone. I don't think. Exact. So as Brittany said, these ANME have never been isolated. Like this is one of those, and who knows how many similar organisms that function in the same way. So ANME are just one example that people detected through analyzing sulfate-reducing bacteria from specific environments. Another fun fact about the sulfur cycle is only part of three largest biogeochemical cycles, which are nitrogen and carbon, and then we add this sulfur cycle. It is highly extensive in the ocean, and there are very important sulfur molecules that are related to the ocean organisms that I will mention now, simply because they are important in so-called global cooling. So I'm not really putting a full name on this DMSP compound because it's something the natural food form it's really complicated to pronounce. But what is important is that microorganisms in the ocean are able to degrade DMSP and to make DMS. So you will see ocean overload with this DMS compound. So why is that important? Because when DMSO gets photooxidized, like oxidized from the sun, it can react with sulfur dioxide, which is SO2, and with sulfate, which is SO4 2-. And they create so-called aerosols. So those aerosols are actually involved in global cooling, which you will agree is really important now when we are struggling with the global warming. So that's again, microbes are those that create this DMS. So they are needed. Yes, it is photooxidation, but microbes degrade DMSP to DMS. Another important alternative carbon source, and sorry, not carbon source. Well, it can be carbon dioxide can be considered carbon source if we think from perspective of carbon dioxide fixation that happens in photoautotrophs or autotrophs generally. But we can also think about carbon dioxide as an alternative terminal electron acceptor. And these are organisms, phototrophs, acetogens, and metanogens that are going to use carbon dioxide as a terminal electron acceptor and as a carbon source. And again, acetogens and metanogens, as well as phototrophs, are first metabolisms that were developed in Earth simply because they could make a use of everything what was available to generate energy. So acetogenesis and methanogenesis, similar but different. So what means acetogenesis? As the name says, acetogenesis is performed by acetogens, which are microbes that generate acetate. Simple as it is. So what is the energy source for them? It's a hydrogen. Simple as it is. Why can be hydrogen? Well, because this was one of the first metabolisms, so only hydrogen was available. It's not phototroph, cannot use hydrogen sulfide, so it's hydrogen. What is respired? Carbon dioxide. Or we can say carbon dioxide is fixed. What is excreted here is acetate, which is why we call them acetogenesis. And the most important enzymes that I will add are hydrogenase and carbon monoxide dehydrogenase. Again, if you are in 311, you would be asked to know all metabolic pathway. This time, I only want you to know hydrogenase, and I will talk more about hydrogenases in special lectures later in class. But with this enzyme, you can take, actually, organism can take electron from hydrogen as a fuel source. And carbon monoxide or CO dehydrogenase is going to use that fixed carbon dioxide and reduce it. And when you reduce carbon dioxide, you get formation of acetate. That's why they're called acetogenesis. Again, don't please memorize the formula of how much energy is excreted. Just memorize hydrogenase and carbon monoxide dehydrogenase. Similar enough is methanogenesis. As you know, it's performed by methanogens. So it's easy to know. Acetogenesis by acetogens, methanogenesis by methanogens. Again, they're different than methanotrophs. They're making methane. Methanotrophs are consuming methane. So same like acetogens, methanogens are using hydrogen as a fuel source, and they're reducing or fixing carbon dioxide. And note that they are in competition with acetogens, and also, as mentioned previously in class, they're in competition with sulfidogens because they all love hydrogen as a fuel source. So they were all one of the first methanogens. So the only difference is that instead of acetate, they generate methane. And again, because I'm not really asking details about metabolism, I'm not asking in details to know the specific enzyme that is called methyl coenzyme A reductase. Principle is the same. Well, not the same, similar, but know only that the difference between them and acetogens is that they make methane, and these components are very significant in anoxic areas like far deep where there is no oxygen because they're full anaerobics. Because when they perform their metabolism, oxidizing hydrogen and fixing and reducing carbon dioxide, they make methane. So if you think about like from the perspective of what's happening in the bottom, so you have a methane generated far down, and that methane just goes up, 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 and they're literally, so there is that barrier on the top, like above methanogens, which are methanotrophs, which are preventing that methane to be released. So they kind of live together. And this is why, but like nowadays, we actually know that one of the biggest contaminants in terms of methane production are actually freshwater lakes and like those, like not rivers, well, rivers as well. Why? Because when there is a lot of sludge accumulated, when you create a lot of anaerobic environment, that is a suitable place for methanogens to develop, and then they just pile up, make a lot of methane, and then methanotrophs that are living on the top just cannot get enough energy to oxidize that methane, and then we get methane released. So this is nowadays actually a big deal, especially in the treatments that are called floating islands, when people are just trying to put a lot of aerobic methanotrophs to prevent those methanogens to release methane. So, and they are here. That's important thing. It was thought that they are bacteria, but they are here. So, what are other electron acceptors that we have? Well, just listing a few, I'm not going to ask all of them, but it's important to know that metals can also be electron acceptors. Iron can be, right? Iron 3+. 2+, iron 2+ can be donor. 3+ can be acceptor. Metalloids, halogenated organic compounds, beautiful. They're really interesting. And many unhalogenated compounds. Metal reduction, iron and manganese. Oh, so sorry. Yes, that's so iron. No, iron can be donor if it's 2+, but it can also be electron acceptor if it's 3+, because when it takes electron, it becomes 2+, because it's more negative, less positive. Manganese, 4+ is reduced to manganese 2+, and I believe that in the next class, not today, but I think next class we're talking a lot about metal respiration, especially about, well, not a lot, but we are going to cover Shevonella and Geobacter, and you will memorize that easily because especially Geobacter is used in something called microbial fuel cell. So it's interesting from biotech perspective. But for now, today, know that they're called metal reducers, and they can use organic source of hydrogen as a fuel source. So pretty much all those anaerobic respirators that are using low energy terminal electron acceptors can use something that is on the top, which is hydrogen or carbons or organic compounds. So what is so cool about Shevonella and Geobacter? Nanowires. So again, when we talk more about metal reducers, you will see how those nanowires are actually helping in energy transfer. So they are literally made of cytochrons, like protein extrusions that are going through membrane and just touch their terminal electron acceptor. So it's called like solid respiration. So I found that very cool. Those cytochrons are beautiful. They're still cytochrons, but they're different than cytochrons that we know in the cell membrane. And last but not least is this proton. And I will never forget the bacteria because I know only this one that respires 8 plus. And I did not know that it exists until I started making presentations for this class. And then I was curious, I was digging in, I was like, this one actually respires protons. How cool is that? To me, it was very cool, especially I love the name. The name is Pyrococcus furiosus. No way that you will ever forget it. What means it's hyperthermophile? That means it lives in a huge, it's archaea, my apologies, it's not bacteria. It can live at 100 degrees. It's like a boiling environment, and this thing actually lives there. So that's where the beauty of the diversity of microorganisms is. It can use sugars and small peptides, but it respires hydrogen. And one day, when I really don't have anything else to do, I will make people in 311 to know the electron transport chain of hydrogen respiration. They are lucky because I really have other things to do. It's not easy pathway. That's why I'm saying it's a lot of enzymes. And for now, we will call it modified glycolytic pathway. But how cool would be to analyze their enzymes for biotech productions? Because think about that. It's actually very highly tolerant to high temperature. What can you do with that? Do a lot. Oh, I can think about some things. So anyway, very interesting. Right? These extreme microorganisms, at least to me, are fascinating. And I guess my excitement, I'm hoping that my excitement will be transferred to all of you because there are so many things that you can research in those extreme environments. So in addition to inorganic compounds, in addition to some halogen compounds and metalloids, there are these inorganic compounds, selenate, arsenate, and tellurate, that can also be used as terminal electron acceptor. So so whenever you see something desulfo, that means it respires sulfate. So desulfo maculatum is respiring arsenate, the poison, and sulfate. That's very cool. Why it's so significant and cool? Because when they respire arsenate and sulfate, so I think they can do both, they generated this mineral. Don't memorize formula, but memorize it. This yellowish color, and you will find it in volcanic areas, in hydrothermal vents, but it's not used anymore because it's unstable. I'm not surprised. And in addition to arsenate, tellurate, and selenate, there are microbes that can respire organic compounds. And again, this just illustrates its diversity. Like literally whatever is there, they will respire. So some of them you maybe know. I mean, don't memorize the full name, but memorize DMSO, memorize TMAO, and memorize these halogenic organic compounds. That's very interesting. Why would be interesting? Because halogen compounds are big contaminants, especially after our waste release. So if you have an organism that can respire this organic compound, that means you can employ it in bioremediation. So this desulfomonyl, dehalococoides, and dehalobacterium, as the name says, dehalo, that means it degrades halogen compounds. So you see, every name has a sense. Like desulfovibrio, desulfomaculatum, peroxidant, that kind of thing. You will memorize that for sure. So they are respiring all of these compounds. So I am never insisting on students knowing, is it chlorobenzene, is it chloroethylene, is it chloromethane? Halogen element is chlorine. So that's what I want you to know, that it's whenever you see that something is dehal, something is expiring, something that is chlorinated, like chloromethane, chlorobenzene. it doesn't matter which compound, but it usually they interconvert, so they can they're not very specific to the carbons, to the respiration. And that would be the last slide for this section, for this part. What we can do